All right, today we're diving into a really fascinating story. It's all about Wilson disease. Now, you might think it's just some rare condition, but it's actually this incredible genetic mystery. It starts with something super simple, the element copper, but it spirals out into a complex puzzle that's literally rewriting what we thought we knew about genetic diseases. So let's start with this question, because it's really at the heart of everything. What happens when something your body absolutely needs turns against you? I mean, we all need copper, right? It's essential for our nerves, our bones, you name it. But what if that very same nutrient started acting like a poison, slowly building up inside you? Well, that's not a hypothetical. That's the reality for people with Wilson disease. Okay, so let's get right into it. The core problem here is a dangerous buildup of copper. You can think of it like this. It's basically a plumbing problem in your body. See, a healthy body is really smart. It takes in the copper it needs and has a system to flush out any extra. But in Wilson disease, that disposal system is just broken. The drain is clogged. So every single day, a little more copper builds up. It starts in the liver, which is kind of like the body's main water treatment plant. And once that's overloaded, it starts spilling out into other places, like the brain. And if we zoom in right down to the cellular level, the damage is pretty dramatic. All that extra copper, it's like lighting a match in a chemical factory. It sets off this chain reaction called oxidative stress, basically a storm of damaging molecules. And guess what gets it first? The mitochondria. You know, the little powerhouses inside every one of our cells. Once they start to go down, the cell's energy just plummets. And that kicks off this whole nasty cycle of inflammation, which can eventually lead to serious, permanent liver scarring, also known as cirrhosis. So why does this whole system fail in the first place? Well, for that, we need to look at the source code, our DNA. It all comes down to one single gene. It's called ATP7B, and it lives on chromosome 13. Now, this gene is basically the blueprint for an amazing little machine, a protein that acts like a tiny, smart pump inside our cells. Its entire job is to grab copper and move it out of the liver. It's the body's master copper regulator, the bouncer at the cellular nightclub, deciding who gets kicked out. And what's really cool is how this thing is built. It's not just some simple blob. It's a complex machine with different parts that all have a job. You've got these domains that act like sensors. They actually find and bind to the copper. Then you have the motor, which grabs onto ATP, that's the cell's fuel, to power the whole thing. There's even a part that provides the spark to get things going, and another that forms the actual tunnel for the copper to exit. It's like a tiny biological factory, and every single part needs to work perfectly. And this is the key. All it takes is for one little part of that machine to break. The sensor could go bad, the motor could fail, the exhaust pipe could get clogged. It doesn't matter which part breaks. The result is always the same. The entire system shuts down. The pump fails. And that's exactly what happens in Wilson's disease. But, and this is where the story takes a really interesting turn, it's not just one single error that can break this pump. We're talking about hundreds of different ways to break it, scattered all over the world. Get this, more than 900. That's how many different mutations scientists have found in that one gene. Think about that. Over 900 distinct ways to break the copper pump, and they all lead to the same disease. You can probably see why this makes diagnosing Wilson disease so tricky. And what's really wild is that these mutations aren't just random. They're like genetic fingerprints that trace the story of human migration. For example, there's one specific mutation that's super common all across Central and Eastern Europe. A totally different one is the main culprit in East Asia. These are what we call founder mutations. It means the genetic typo probably happened just once, a long, long time ago in a small group of people, and then it got passed down through generations as populations expanded. It's like history written in our DNA. Okay, so all of this brings us to the real heart of the mystery. For decades, scientists and doctors thought they had a pretty good handle on how many people had Wilson disease, but it turns out they were way off. So for the longest time, if you opened a medical textbook, you'd see this number, 1 in 30,000. That's how common Wilson disease was thought to be. And that was based on a pretty simple metric, counting the patients who actually walked into a clinic and got diagnosed. Seems simple enough. But then genomics happened. We got really, really good at reading DNA. And as scientists started sequencing the genes of thousands upon thousands of everyday people, they found something that just blew their minds. The number of people who actually had the genetic recipe for Wilson's disease, 2 bad copies of that ATP7B gene, it wasn't 1 in 30,000. Nope, it was more like 1 in 7,000. I mean, just look at that gap. It's not a tiny little difference. We're talking about a massive, nearly 5 times difference between the number of people who, genetically speaking, should have the disease and the number of people who actually get diagnosed. It's huge. So, that leads to the million-dollar question, the one researchers have been obsessed with. Where are all the missing patients? Are they out there, just undiagnosed, or is something else going on? How do you explain a gap that big?
Well, the answer is it's complicated. The story is so much bigger than just that one broken gene.
This has led to a new way of thinking called the multi-hit hypothesis. The idea is simple. Having those 2 bad copies of the ATP7B gene, that's just the first hit. It's a big one, for sure, but it might not be a knockout punch on its own. Whether someone actually gets sick and how sick they get depends on other hits from other genes, from your environment, maybe even from epigenetics, which is all about how your body chooses to read its own genetic instruction book.
Think of it like a movie. The ATP7B gene is the main character, but there's a whole supporting cast of other genes. And these modifier genes can totally change the story. Some of them might be protective, helping you out. Others can make things way, way worse. This helps us finally understand that really baffling question. How can 2 siblings who both have the exact same primary mutations for Wilson disease have totally different outcomes? One might get really sick, while the other has barely any symptoms. It's because their genetic supporting cast is different. It's a whole network effect.
So, figuring all this out isn't just for a bunch of scientists in a lab. All this knowledge is leading to a complete revolution in how we actually treat Wilson's disease. For so long, the treatment was just management. You'd take drugs every single day for the rest of your life to either pull copper out of your system or block it from being absorbed. It helps, but it doesn't fix the core problem. But now, we're on the verge of something completely different. We're talking about potential one-time genetic cures, therapies that go right to the source and fix the broken gene itself. And listen, this isn't some far-off sci-fi dream. This is happening right now, in clinical trials. There are a few amazing approaches. One is called gene addition. They use a harmless virus, like a delivery truck, to drop off a correct, working copy of the gene in your cells. Another uses mRNA technology. Sound familiar? Yeah, just like some of the COVID vaccines, to give your cells the temporary recipe to make the right protein. And then there's the holy grail, prime editing. It's like a super precise version of CRISPR that can literally act like a find and replace function on your DNA, correcting the typo permanently. It's a genuine genetic fix. So when you step back, the story of Wilson disease is about so much more than just copper. It's really a glimpse into the future of medicine. We had a disease that we were convinced was rare, but it was actually hiding right under our noses just because it was more complex than we realized. And it makes you wonder, doesn't it? As we get better and better at reading and even rewriting our own genetic code, how many other so-called rare diseases are out there, just waiting to be discovered, hiding in plain sight? And how many of them might be more curable than we ever dared to imagine?